Hi everyone, this is our separation processes project on dialysis. Damn it. Oh, it's the mouse.
Okay, so let's start with the basics. Dialysis is a way to separate small particles from larger ones using something called a semi-permeable membrane. This just means that the membrane lets some stuff go through, like small particles, but keep the bigger ones out. And today, most of us think of dialysis as a medical treatment for people with kidney problems, right? But actually, it started out as a science experiment back in the 1800s. Over time, it turned into this life-saving medical technology that we have now. So the first person to really study dialysis was Thomas Graham, a Scottish chemist. This was back in 1861. He was the one who gave dialysis its name. He discovered that if you put a solution next to a semi-permeable membrane, small molecules like sugar or salt could pass through. That's what he called crystalloids. But bigger molecules like proteins stayed behind. That's what he called colloids. At that time, his work was for chemistry labs. Nobody thought about using it for medicine yet. But his research set the stage for everything that came later. In the early 1900s, some scientists started testing dialysis on living things. In 1913, John Abel and his team in the US used dialysis on animals to try to clean their blood. It was a pretty big step, and it showed that maybe this could work in some medicine someday. In 1924, George Haas was the first person to actually try dialysis on a human. It didn't really work, but it still was the first time. And that matters because it helped open the door to future experiments. So now, here where's the thing get exciting. During World War II, a Dutch doctor named Will Kolff built the first working dialysis machine, basically an artificially kidney. He used super basic materials like sausage casings and parts from a washing machine. In 1945, he used it to treat a patient with kidney failure, and it worked. And this is when the modern hemodialysis started. Then, in 1960, an American doctor named Belgin Scriver took it even further. He created a device called the Scribner shunt, which made it possible for patients to get regular dialysis without surgery. Thanks to him, the first dialysis clinic opened and long-term treatment became a real option. So today, we have two main types of dialysis. We have hemodialysis, where your blood gets cleaned outside of your body using a machine, and peritoneal dialysis, where your own blood, specifically the lining of your belly, filters the blood inside you. So technology has come a long way since the sausage casing days. Machines are way more comfortable and efficient now, and researchers are even working on wearable dialysis devices, so patients can have more freedom in the future. So yeah, dialysis started as a chemistry idea, and now it's something that literally saves lives every day. And it's a cool example of how science can go from a lab experiment to something that changes people's lives around the world.
Okay, next we're going to get into some of the industrial applications of dialysis. Dialysis is a membrane-based separation technique that utilizes semi-permeable membranes to separate solids based on their size and charge. In industrial contexts, dialysis is employed for tasks such as metal ion recovery, acid reclamation, and sample preparation for analytical procedures. One example of this is Dannon dialysis in the industry. Donin dialysis is also known as diffusion dialysis, and it operates on the principle of ion exchange across a membrane driven by concentration gradients. This method is particularly effective for metal ion recovery, as it facilitates the enrichment and separation of metal ions, including the recovery of noble metals like gold, copper, and silver from dilute solutions. And also in acid recovery, Donin dialysis enables the reclamation of acids from solutions contaminated with metal ions, allowing for the reuse of acids in industrial processes. The process is energy efficient as it does not require external energy inputs, making it cost effective for large scale applications. And in the photo over here, this schematic is illustrating a Donin dialysis cell. It showcases the arrangement of cation exchange membranes and the flow of ions between compartments. It effectively demonstrates the principle of ion separation driven by the concentration gradients across ion exchange membranes. Okay, so applications in the electroplating and metal processing industry, kind of going deeper into metal acid recovery, where we're looking more into how this is effective in the electroplating industry. So dialysis is used to recover metals such as zinc and copper from rinsed solutions. It also separates mixed acids like hydrofluoric and nitric acid from spent leaching solutions. And it can reclaim sulfuric acid from waste streams containing metals like iron and nickel. And these applications don't not only reduce waste, but also allow for the recycling of valuable materials. Looking more to the figure, the schematic represents the recovery of acids through diffusion dialysis, highlighting the use of anion exchange membranes to separate and reclaim acids from industrial effluents containing dissolved metals. It is particularly relevant to processes in electroplating and metal finishing industries. Finally, getting into more analytical and environmental use, dialysis is also employed in analytical chemistry for sample preparation. Two examples of this are flow injection analysis and sequential injection analysis. Starting with flow injection analysis, or FIA, dialysis membranes are integrated into FIA systems to remove interfering substances, enabling accurate analysis of components like phosphates and chlorides in complex matrices. For sequential injection analysis, or SIA, dialysis aids in the on-line removal of suspended solids and the dilution of analytes, improving the precision and efficiency of analytical measurements. These techniques enhance the reliability of analytical results while minimizing sample contamination and reagent consumption. The diagram on the right depicts a flow injection analysis system, which is used for the determination of hydronium ions. It illustrates the integration of dialysis and analytical chemistry setups to remove interferences and suspended solids, thereby enhancing the accuracy and reliability of chemical measurements. In conclusion, dialysis serves as a versatile and efficient method for separation and recovery in various industrial processes. Its applications in metal recovery, acid reclamation, and analytical sample preparation highlight its significance in promoting sustainable and cost-effective industrial practices.
Okay, so now I'm going to get and talk a little bit more about the different processes of how dialysis works. So first, we want to talk about the main principle behind dialysis, which is osmosis. And so osmosis is the spontaneous diffusion of water or other solvents through a semi-permeable membrane. A semi-permeable membrane is a membrane that blocks the passage of certain dissolved substances, but allows the passage of other smaller molecules and water, or the solvent that's used. And it naturally moves solvents across a membrane from the side of higher concentration to lower concentration. Reverse osmosis would be the reverse of that process. So it would move it from a lower concentration to a higher concentration. And usually this is done under pressure to do this. And so it results in filtration of a solute from the solvent and leaving behind the solutes. And this is what is exemplified in this picture. So if you look at the top picture, we have the sugar and the water molecules. There are less water molecules on the left side, but there are more water molecules on the right side. And so the water is going to move from a high concentration to a low concentration, therefore diluting the right side. So osmosis in dialysis. So blood contains many substances that are dissolved in particles. And so during osmosis, you don't want the blood cells to be removed from the blood. You just want excess water. And so in dialysis, it is brought very close the blood is brought very close to a clear liquid called the dialysate that contains few particles. The blood and the dialysate are separated only by a thin wall called, again, a semi-permeable membrane. And as I said, it doesn't let important parts of the blood, such as the blood cells, to pass through it. The dialysis fluid or the dialysate contains sugar, which helps to draw water out of the blood. Some methods use pressure to push water out of the blood. When the water is removed, any harmful substances that can be dissolved in that water are removed as well at the same time. And this process is called convection. So in this picture, this is an example of a type of dialysis that we will discuss later called hemodialysis. And so this is done outside of the body. The clean blood goes into the dialysis machine. The dialysate and the blood are run counter current to each other. And the dialysis fluid is going to always be replaced. So as mentioned in the last slide, one type of dialysis is called hemodialysis. In this process, the patient's blood is removed from the body and pumped into the dialysis machine, and then is returned to the body. This is called an extracorporeal process. The dialysis fluid flows countercurrent to the blood, as was mentioned previous, and this takes about 4 to 5 hours. In the machine, the blood flows in tiny tubes surrounded by the dialysate, and the walls of the tube act as a semi-permeable membrane that allows only small molecules to cross it. These include molecules such as nitrolysis waste, water, and electrolytes. Filtration occurs through osmosis and diffusion, where the water molecules and solutes move from a higher concentration, which would be in the blood, to a lower concentration in the dialysate. The dialysate contains solute levels that are similar to the levels in healthy blood. Other substances can also be added to the dialysate to give to the patient. These can include bicarbonate, erythropoietin, and other medications. And then the second type of dialysis is peritoneal dialysis. This dialysis, the dialysis fluid is entered into the abdomen, and the lining of the abdomen, which is called the peritoneum, serves as a natural semi-permeable membrane. And so over time, the blood will be cleansed of any excess water through that membrane. The dialysate contains excess sugar, which removes the excess water from the blood by osmosis. And then the fluids typically remain in the body until an equilibrium is established with the fluids in the blood. One of the advantages of this process is that it's more flexible, it's more tolerated, it's less expensive, and it takes about 8 to 12 hours, which could happen while the patient is sleeping.
Hi, I'm going to be talking about the mathematical derivation of dialysis. So first of all, I'm going to start with something that we already looked at in class. Here I'm drawing the film, and as you can see, this is going to be the depth of the membrane right here. So unlike other types of transport in class, this membrane actually has a depth. So here is going to be the blood, and here is the dialysate on this side. So on this axis right here, this is concentration. So that is concentration. And then I'm going to start drawing the different concentrations. So right here is the concentration of the blood. That's similar to what we've seen in class. And then the concentration of the dialysate. The differing thing that we have here is the line in the membrane. So these represent the mass transfer coefficients. So in blood, the mass transfer coefficient is going to be Kb. In the membrane, it's going to be Km. And in the dialysate, it's going to be Kd. To get the overall mass transfer coefficient, we have 1 over K, which will be the overall mass transfer coefficient, and that's going to equal 1 over Kb plus 1 over Km plus 1 over Kd. So with the overall mass transfer coefficient, we can then determine the efficiency of the dialyzer. So first of all, we have to calculate the number of transfer units, and this equation can be found in our textbook. So we have K, which is our overall mass transfer coefficient, A, which is the area of the membrane, and that's going to be over QB, which is the volumetric flow rate of the blood. So in order to define the efficiency, which we're going to call E, that's going to equal CBI, which is the concentration at the blood inlet, minus CBO, which is the concentration at the blood outlet, over CBI, again, the blood outlet concentration, minus CDI, which is the inlet dialysate concentration. All right, so as we know, in the dialysis machines, it's usually not using water. So I'm gonna talk a little bit about the difference between water and blood. So we're gonna call that clearance, which we're gonna use as CL. So that's gonna equal 1 minus CPO over CPI, which is the ratio between the plasma concentration at the outlet, which is CPO, over the plasma concentration at the inlet. And we can multiply that by QB, which again is the blood flow rate. So this would be used in the equation if we were using blood as opposed to water. So next, we're going to talk about a little bit of transport. So we're going to bring back our favorite flux equation. So we have J times C equals D dC dy. J is the flux, C is the concentration of the solute, D is the diffusion coefficient, and Y is the distance from the membrane. If we integrate this equation, we have J equals D over lowercase delta. And lowercase delta is the thickness of the membrane. And we're going to multiply that by ln CW over CB. And CW is the concentration of the solute, and the CB is the concentration of the bulk. So that's going to be with the solute moving through the membrane, if the solute can pass through the membrane, which we have talked about. Since we're not only dialyzing water, you have to take this into account. So the last part mathematically I'm going to talk about is the dialysence, which is DD, is how I'm going to describe that. So that equals MI dot, and that is the mass transfer rate over CBI minus CD. So CBI is the concentration at the blood inlet, and CD is the concentration of the dialysis inlet. Since we're only using the dialysis inlet, we can drop the I because it's unnecessary. So as I said before, MI is the mass transfer rate. So MI equals QB, which again is the blood flow, and that's going to be CBI minus CBO, which again, concentration of the blood outlet minus the concentration of the blood inlet. Using this equation and a couple derivations, we have MI equals AU CBI minus CD, just like before, minus CBO minus CD. So basically, this is the same, this top line is the same as this over here. We're just adding the CD terms. And then since we have A times U, that's QB, but you also have to add this ln here at the bottom so that we have our logarithmic mean equation right here. So after that, we can solve a little bit more. We can reduce it just a little bit more. So we have QB times CBI minus CD. And then using this past equation, we have 1 minus the exponential of negative AU over QB. And subbing that into our dialysate equation, we have DD, which is the dialysate, QB, which is the blood flow rate, 1 minus the exponential of negative AU over QB. And that is our final equation to solve for dialysate, which once again is an indicator of solute removal performance of the dialyzer. So not only did we discuss dialysate, which can be used to optimize the dialysis process, we also talked about the fluid flow and flux through the dialysis process, and we talked about the efficiency of the dialysis process, and we also used a little bit of our knowledge from class from our concentration plots. So I hope that makes sense.
There we go. Okay, so now we're going to be looking at the future of medical dialysis. First, looking at it from an epidemiological perspective. So, unfortunately, currently, millions of people die of kidney failure each year because of lack of access to kidney replacement therapy, which includes dialysis, both hemodialysis and peritoneal dialysis that we discussed earlier. As we can see in the second figure, the number of people worldwide estimated to need kidney replacement therapy is outpacing the number of people who will receive it. As you can see, the slopes of the dark blue line is greater than the light blue line. This is a big problem and very unfortunate. The prevalence of dialysis use in a country is also strongly correlated with the national income, but interestingly, the rate of growth of dialysis use is larger in poorer countries as dialysis becomes more accessible. Yay. Okay, kidney failure is likely becoming more prevalent now and in the future for a variety of reasons, including the current aging global population and the increasing prevalence of key risk factors such as diabetes and hypertension. So, as a result, total dialysis costs across the population are expected to rise as medical therapeutics and kidney failure causes like diabetes have gotten better. So, which actually increases the lifespan of the patients, but also increases the time each patient will spend on dialysis. So, I guess it's kind of a give and take in that sense. Because right now, kidney replacement therapy absorbs about 5 to 7% of the total healthcare budgets, even though kidney failure affects only about 0.1 to 0.2% of the general population in most regions. So as you can see, it's like the cost is not necessarily outpacing also the prevalence per population. So now let's look at dialysis, the future of dialysis from a research perspective. So as kind of mentioned earlier, current shortcomings of dialysis include the cost, which includes like the cost of manufacturing and the treatment, and relatively bad outcomes, which specifically means lower survival rates for kidney failure compared to kidney transplantation and even some other cancers, and incomplete restoration of kidney function, specifically meaning it does not restore metabolic activities in the kidney. It does not restore like hormone production. It does not help in blood pressure regulation, or the tubular reabsorption of essential substances like glucose and amino acids. Also, dialysis has a large environmental burden. Specifically, it's about 20 times higher than kidney transplantation, specifically due to water consumption in the production and the use, the greenhouse gas emissions, and waste production. And also, dialysis has a pretty heavy, significant impact on the quality of life of the patient with very distressing symptoms, which include itching, cognitive dysfunction, pain, loss of appetite, and obviously restrictions in social deployment because of the time that the treatment takes. So here's some new ideas in research that are gonna hopefully going to help these patients and solve some of these problems. So one of the first interesting ideas I think is the idea of regenerative medicine, specifically growing a kidney out of the patient's stem cells, as opposed to traditional kidney transplantation. And since the new kidney would contain the DNA of the patient, there is a very little chance that it'll be rejected, which is a big problem in traditional kidney transplants. However, currently research in these stem cell grown kidneys show limited filtration function and have not yet been generated for human application. Yeah, currently stem grown kidneys have shown pretty limited filtration function and have only been, I guess, like used in small animal models and have not yet been generated for human application. Currently, what seems like to be the more viable option is to use the patient's stem cells to repair the kidneys instead of completely replacing them. Okay, so another interesting point of research is the bioartificial dialyzers, which utilize living tubular cells that are seated on the dialysis membranes. So this advancement will be useful to remove uremic solutes more efficiently by causing an extra solute shift from blood to dialysate via the transport systems in tubular cells, specifically, especially for the protein-bound toxins, which are way less efficiently removed by hemodialysis. However, currently, uh, some constraints for this approach include scaling and finding a way to keep the cells sufficiently functional over time, because we got to keep them alive. And then next we have adsorption. So currently, some sorbents are also used to directly remove solutes from blood or dialysate. They contact with the blood via beads in cartridges or that are seated on dialysis membranes or applied for dialysate regeneration and reduction of dialysate volume. So currently, future research aims to make the sorbents reusable and to find sorbents that can aid in urea removal. And next we have green dialysis. So as I mentioned earlier, dialysis production and use has a pretty big effect on the environment. So drastic changes are needed to make the current dialysis concept energy neutral and recyclable, particularly in its production. So steps include the reduction of water consumption by dialysate regeneration and using the reverse osmosis reject, which is part of the manufacturing process for a second purpose. So basically just like using the water for more than one purpose and making material production and the dialysis process itself energy neutral. And of course, recycling plastic waste. So the final, I guess like industrial exciting future of dialysis is in electrodialysis, which is basically just dialysis in under the influence of a current, and currently has lots of potential for wastewater treatment, where its properties like its selectivity, the high separation efficiency, and chemical-free treatment make it really good for desalination and other treatments with significant environmental benefits. So these electrodialysis technologies can be used in operations of concentration, dilution, desalination, regeneration, and valorization to reclaim wastewater and recover water and or other products that you would like heavy metal ions, salts, acids and bases, nutrients, and organics, or even electrical energy. So yes, very exciting. Figure, this would be an example one of the bioartificial dialyzers as treatment inside a person to replace, I guess not to replace, but to aid in kidney function. There are sources, and yeah. Thank you all very much for listening to our presentation on dialysis, and yeah.